Today we're going to talk about the biochemistry of nervous system. And first of all, we're going to talk about the blood-brain barrier. As we know, in the brain we have blood-brain barrier, which it allows movement of some materials from the blood to the brain while restricting other materials to go to the brain. And the materials which are allowed to go are the first fuels, and second the amino acid, third the vitamins. First, the fuel. Brain uses glucose as a fuel, and it's very important because the brain does not have glucose as stored form, and it uses directly from the blood through the way of GLUT1 through facilitated diffusion. And glucose lower than 60 mg per deciliter accounts for hypoglycemia. But in cases of starvation, when there is no glucose, the body will use the ketone body as source of fuels, as they can pass through the blood-brain barrier as well. We have the amino acids, second thing which crosses. Amino acids are important because they act as precursor for synthesis of proteins and for the synthesis of neurotransmitters. They co-work with the vitamins, which can cross as well, to produce the neurotransmitters. The amino acids, there are two types: long neutral amino acids and small neutral amino acids. Small neutral amino acids, their movement through the blood-brain barrier is restricted because they will interact with the formation of neurotransmitters. So the long neutral, which they pass easily, are divided into essential, which are the phenylalanine, isoleucine, leucine, valine, methylene, tryptophan. And while in non-essential we have tyrosine, and semi-essential is the histidine. Small neutral non-essential are the alanine, glycine, proline. These which are movements are restricted. So now we're going to talk about the general properties of neurotransmitters. Neurotransmitters are synthesized and then they are stored at the vesicles at the presynaptic. This is the axon, this is the axon bulb, the presynaptic, and then this is the synaptic cleft, this is the postsynaptic. So the neurotransmitters which have precursors, for example the amino acids are the precursors, and the vitamins as well, they work together to produce the neurotransmitters. These neurotransmitters are then stored at the vesicles, and when an action potential comes, it induces, this vesicle will go under exocytosis, it will bind to the membrane, and then the neurotransmitter will be released to the synaptic cleft, and then after that it will bind to the receptor, initiating a response at the postsynaptic cleft. And then after that, this neurotransmitter will undergo removal by two ways: either by degradation through enzyme, for example acetylcholinesterase, or by endocytosis. By endocytosis I mean the reuptake of the neurotransmitter by the vesicle, by the empty vesicle, again to use it again. We have two types of neurotransmitters. We have the excitatory neurotransmitters and the inhibitory. The excitatory, for example, we have acetylcholine, aspartate, dopamine, norepinephrine, epinephrine, histamine, glutamate, serotonin. And the inhibitory, we have the GABA, gamma-aminobutyric acid, and the glycine. So this was general properties of neurotransmitters. And now we're going to talk about the synthesis of acetylcholine. Acetylcholine are synthesized from choline and acetyl-CoA by the help of an enzyme which is called acetyltransferase. We'll have acetylcholine. Then, after this acetylcholine been synthesized, it will be stored inside these vesicles until the time to be released. When an action potential comes along the axon, it reaches the presynaptic cleft, and then there will be a change in the membrane of the presynaptic cleft, and the calcium ions will enter through the membrane, and inside there will be increase in the level of calcium, and this will cause the vesicles to be bound to the membrane of the presynaptic cleft, causing exocytosis, and these acetylcholine will be released into the synaptic cleft. And then after that, this acetylcholine will be bound to the receptors. We have 4 types of acetylcholine receptors. We have alpha 2, beta, the gamma, and then the delta. And it will bind to the specific receptor, initiating a change in the postsynaptic lift. And these membranes usually do not allow diffusion of ions from one side to the other. But when the acetylcholine binds to the receptor, it causes sodium ions to come inside and the potassium ions to go outside. While inside there will be increase in the level of sodium ion, it will cause depolarization, and then the action potential will continue along the axon. These neurotransmitters, after they induce the action, they will be rapidly removed by 2 processes, or by degradation through the enzyme, which is called acetylcholinesterase, or by endocytosis as well, the reuptake of the neurotransmitter. Now we finished with the acetylcholine. We'll talk about the catecholamine, another neurotransmitter. Catecholamines, for example, dopamine, epinephrine, norepinephrine. The formation of these 3 include 4 steps. The first step, which is the hydroxylation. What we need in this step, we need the tyrosine oxygen tetrahydrobiopterin. These 3, with the help of the enzyme called tyrosine hydroxylase, will change into dopa and dihydropterin. What is important in here, the dopa, because we're going to use it in the next step. In the second step, which is the decarboxylation, the dopa which we had in the first step will change into dopamine by the help of an enzyme called dopa decarboxylase, which deficiency in this enzyme causes Parkinson's disease. So in the second step, we had the formation of dopamine. And now we'll go to the third step. In the third step, which is hydroxylation, the dopamine, which we had already synthesized with the oxygen and vitamin C ascorbate, will change to norepinephrine, water, and dehydroascorbate by the help of an enzyme called dopamine beta hydroxylase. You see, dopamine beta hydroxylase. Third step, which is the hydroxylation, will change the dopamine with the vitamin C and O2 to norepinephrine, water, dihydroascorbate. The final and the fourth step, which is the methylation. In this step, what we have, the norepinephrine, which we produce in the third step with a methionine amino acid, will change into norepinephrine. And the importance is that the enzyme, which is called phenylphthalo-N-methyltransferase, will change S-adenosylmethionine with norepinephrine to epinephrine and S-adenyl homocysteine. The important in this, the norepinephrine with the methionine amino acid will change into epinephrine by the help of phenylphthalo-amine N-methyltransferase. So this was the 4 steps of the formation of catecholamines. Now we will talk about the formation of serotonin synthesis. Serotonin is synthesized from the amino acid. It's composed of two steps, and it comes from the tryptophan amino acid. And at the beginning, we have the first step. We have the tryptophan, which will undergo hydroxylation by the enzyme tryptophan hydroxylase. We'll have 5-hydroxytryptophan. And in the second step, which is the decarboxylation, the 5-hydroxytryptophan will undergo decarboxylation to become serotonin, which we need by the enzyme hydroxytryptophan decarboxylase. Hydroxytryptophan decarboxylase. This is a step called decarboxylation. Okay. And the removal of serotonin is by 2 processes, or by oxidation or dehydrogenation. Oxidation by monoamine oxidase and dehydrogenation by aldehyde dehydrogenesis. So this was a simple review of formation of some of the amino acids